Welcome to our lab podcast on the female reproductive system. Here we are going to walk through and look at additional images that are going to support what we learned in our lecture podcast about the female reproductive system. So here we have a low magnification view of cross section of a female ovary. We're going to have the capsule out here. First, remember we've got our germinal epithelium, that columnar type or high cuboid type epithelium, the germinal epithelium with the tunica albuginea beneath. All of the white spots we see in the cortex, these are going to be our primordial follicles. Then as we move through follicular development, we can see a number of these in increasing size. Here we're approaching what could be a Graafian follicle. We still have it attached, so I would technically still call this a secondary follicle. But you can see how large some of these get. And there are a number of these, and this really supports the fact that throughout follicular development, a number of follicles develop, but usually only one is lost to ovulation. Looking a little bit closer, we can see our germinal epithelium at the surface. This is cuboid shaped in nature. We have the tunica albuginea, our connective tissue. Then we have our first type of follicle. This is going to be our primordial follicle. Mostly what we're looking at is oocyte. And we've got our squamous single layer follicular cell surrounding. Here we can see a nice view of the oocyte. This is the oocyte nucleus. And so this is going to be the germinal body here of our oocyte with our squamous follicular cells. So here we have a primordial follicle. Now, pulling away just a little bit, we can see once again where we have our primordial follicles in this outer cortical region. Then we have our number of different types of stages of follicular development as we move along. This will be our Graafian follicle. You can see almost completely detached. Most people will go ahead and call this Graafian, but really for technical reasons, if there's still an attachment, then we still call it a secondary follicle. This is one of the images that we saw from lecture. This is a perfect example of a multi-laminar primary follicle. Here you can see the multiple layers of our granulosal cells, zona pellucida, and then our germinal portion, the nucleus of our oocyte. Outside we have our theca interna cells, and we have our stromal type theca externa cells. Now, this is going to be our unilaminar primary follicle. Here we can see a single layer with our zona pellucida, with our oocyte, beginning to develop some of our theca cells on the outside. But here is a primary unilaminar follicle, also probably unilaminar, even though we don't necessarily see the nucleus in these. But you can see how these are more cuboid to columnar in size. Certainly they are larger than our primordial follicles. This is an example of an atretic follicle. This is pretty easy. We can see all of these really dark pycnotic nuclei, but as these granulosal cells are detaching and free floating within the antrum in the liquor folliculi, that's also another example of the degenerating atretic follicle. Here we have some follicles in different stages as well. We can see our primordials along the surface, but what we're looking at, especially here, this is going to be our secondary follicle. We can see these spaces where we're filling with liquor folliculi. We're disrupting the granulosal cells in our multiple layers. So this is going to be our secondary follicle. I can't tell if we have one or two at this magnification. If that is a single layer of follicular cells, then that's still a primary unilaminar follicle. This is likely going to be a secondary follicle, maybe even a Graafian follicle. We just don't see the oocyte in this plane of section, which also might mean that we're looking at an atretic follicle. I don't really see the pycnotic nuclei. We just don't have enough information here to really help us out a lot. Here, we're probably going to go with, I personally would go with secondary, but they're probably going to say Graafian follicle because of how large this unit is. But here we have the oocyte, we have our corona radiata cells, cumuloso-ophorus covering the corona radiata and creating the bridge back to the granulosal cells. Personally, since there's still such a big attachment, I will go with secondary follicle for this one. This is just a higher magnification look at the follicles that we saw before. Again, the antrum. Many of these spaces are going to coalesce into the single large antrum, so secondary follicle. Here we have one layer, so we can see that a little bit better. So that is a unilaminar primary follicle. And we don't see the oocyte present here, but since we have this large fluid-filled space, then it's going to be a secondary as well. Here we're just looking at a higher magnification of the oocyte. I refer to it as an oocyte unit that's present suspended within the antrum and the liquor folliculi. Here we can see the oocyte, corona radiata, then all of these cells surrounding and attaching back. These are the cumuloso-ophorus cells. Here we have our granulosal cells. So probably because we've just practically detached here, we will go ahead and go with Graafian follicle for this one. Now, as we begin to look at the cells of the periphery, here we have our antrum filled with liquor folliculi, our granulosal cells. These are going to be our theca interna cells, and you can see the number of red blood cells in here. This is highly vascular. You can see the vesicles of the secretory nature of the theca interna cells, and then we've got our stromal type theca externa present on the periphery here. Here we have examples of follicles quite easily on the right. We can see the detachment of our granulosal cells, some deep, dark, small spots. These are going to be our atretic follicles, and these are our pycnotic nuclei that are present here. Over here we can see a little bit of detachment. This one is a little bit further along, but we either have an atretic follicle or we have a follicle where we have the oocyte out of the plane of section. This would be more typical of what I would show you for an atretic follicle. You'll definitely have more cells like this. This one's a little bit more challenging, although I would still go with an atretic follicle here as well. A little bit closer magnification, again, you can see the really, really small, dark nuclei that are present. Those are the pycnotic nuclei that you have there, and then you can see the shedding and the detaching of the granulosal cells. Here we have another example of an atretic follicle. This helps you distinguish even better the pycnotic nuclei, the really dark, dark spots that you see here. And again, you can see the loosening of the granulosal cells. Here we have an atretic follicle. Here we have a multilaminar follicle. This is primary. We don't see fluid-filled spaces. Here we see a multilaminar primary follicle. Again, thick zona pellucida surrounding the oocyte. We got a thinner zona pellucida around this smaller one. Here we've got a zona pellucida and some granulosal cells, but we've lost the oocyte. The oocyte is no longer present in this particular one. Now, as we look in the medullary portion of our ovary, we're going to be looking at a number of different types of structures. Here we have a corpus albicans. This is going to be more of the connective tissue portion, the portion that is lost from the scarring that occurs after the corpus luteum degenerates. We also have a smaller one that we can see here, but this is a corpus albicans. In this one, again, we're looking at a corpus albicans. We don't really see the highly folded nature that's typical of what we see with a corpus luteum, and in that case, a lot more stromal tissue type material that we see here. And so quite small in nature as well. So we're going to go with corpus albicans for this particular one. Here we can see a section through the ovary, and we have two very large corpus lutea. These are in different stages of development. Here we can see the folded nature of this corpus luteum, but you can see how large this corpus luteum is, how it's filled with these granulosal cells that are just chugging out the progesterone to maintain the endometrial lining for about those 2 weeks period of time before menses would occur. Again, just looking at sections through the ovary, here we have a much later corpus albicans, probably, corpus luteum moving into an albicans here. We don't see the folded nature of that. So this one's much older. We can see some of the folds here on the lateral sides, but this one's probably a very old corpus luteum at best as it's beginning to regress. Here we see some very large cyst-type structures. These are going to be Graafian follicles that are regressing as well. This is fairly typical of what we see with a corpus lutea. You can see these granulosal cells all folded in nature. Here we're going to have some connective tissue septa that help divide these granulosal theca cells, granulosal lutein cells, into these folded structures. This is going to be the lumen, and here we'll have our theca lutein cells on the outside. Again, here we're looking at the folded nature of a corpus luteum. And as we're looking at these, we can see the connective tissue septum that's driving up. This is going to be our theca lutein cells, formerly the theca interna cells, as they're projecting up. These are going to be, again, secretory in nature. And then here we have our granulosal cells that are the highly folded nature. And again, you can see the secretory vesicles present in these folds. These, again, are our progesterone factories. And again, just a higher magnification look at the connective tissue septa that we see at the bottom, and then the granulosal lutein cells that are present in the corpus luteum. Here we have a section through the infundibulum, or the end of our oviduct, and we can also see these sections of these finger-like processes called the fimbria extending away from the infundibulum. Here we have the opening, or section through the opening, and all of these curled ribbon-like structures that we see. These are the fimbria portion of the infundibulum of the oviduct. We can see the highly folded nature. This is not uncharacteristic of what we're going to see in the oviduct. And in fact, we can judge a little bit of the depth within the oviduct. As we're at the infundibulum and the ampulla, the smooth muscle of the muscularis is going to be thinner and the mucosal much thicker. And as we move closer to the uterus, that's going to change. The mucosal layer is going to become thinner and the muscular layer is going to increase in its size. So that's one way that we can distinguish if we're at the ampulla end or if we're at the intramuscular end or somewhere in the middle in the isthmus of the oviduct. Here we can see the epithelium of this folded nature. We can have some ciliated cells, as we see, and then we have non-ciliated cells that are present. These are going to be secretory in nature. We'll see better examples of the peg-type cells that we see, but here we can definitely make out the ciliated versus the non-ciliated cells of the mucosa of the oviduct. So here we're moving into the deeper regions. This will be in the ampulla region. We can see the mucosa versus the muscularis. This would be the thickness of the mucosal layer, and there we have the muscularis layer surrounded by our serosal layer. Here we have some of the ligaments it's extending away. As we look more closer at this highly folded nature, they call these infoldings placae. You remember the placae circularis that we had in the gastrointestinal tract. These folds are just placae, and you can see they're quite angular in nature, and that's one way to help us distinguish where we're located in the female reproductive tract. As we look a little bit more closely, we can get an appreciation for the epithelium that is present on the surface of these placae. Even at this lower magnification, we can appreciate that we have these large bulbous cells projecting out on the surface intermixed with ciliated cells. So these are going to be our peg cells. And at closer magnification, they're even more evident as we see the pegs rising very highly above their neighboring ciliated cells. So these are the peg cells, secretory in nature, providing nutritive and protective support for the spermatozoa. And here we have the ciliated columnar cells that are creating the current toward the uterus itself. Now, getting ever closer to the uterus, here we can see a fairly thin mucosa, very thick muscularis. So we are in the isthmus and approaching the intramuscular portion of the oviduct. Now here we have a section through the uterus. We can recognize the very dense red myometrium along the bottom versus the endometrium with all of its uterine glands streaming through. This will be the basalis layer, and the majority of the endometrium we're looking at is the functionalis layer that's going to be lost every month during menses. Here we have a closer look. Again, we can see the smooth muscle of the myometrium curving around in this direction, and then the majority of these white spaces are going to be our uterine glands. As we look in other sections, we're going to begin to recognize our straight and spiraling arteries and veins that are present in the endometrium. The straight vessels will be near the basal portion, whereas the spiraling arteries will be closer towards the superficial, or definitely in the functionalis portion. Once again, looking at the endometrium, we have our surface mucosal cells invaginating into our uterine glands. And here we begin to see some of our vessels, and if we are this near the surface that we see here, our simple columnar epithelium, here we can see we're diving down into an endometrial gland. These are going to be portions of our spiraling arteries that are very near the surface. Here we just have the lining of a uterine gland, simple columnar epithelium, quite a lot of material. And we can see a lot of the storage of material that's going to be present here, not the least of which in these uterine glands is going to be glycogen. Again, material to help nourish the developing zygote until the placenta has been adequately formed. Here we're looking at the base of a uterine gland, and we can see that now we have fairly long longitudinal sections through blood vessels This is going to tell us we're getting down toward the basal layer, and these are the straight portions of our arteries and veins present in the endometrium. And lastly, although this is a low magnification view, we're looking at stratified squamous paraceratinized epithelium. So we're looking in the vaginal canal. We can see a little bit of a section through a muscle bundle here. This is going to be sort of overlapping circumferential and longitudinal muscle bundles that are present out here in the muscularis of the vaginal wall. And again, a closer magnification, fairly typical of what we see with stratified squamous. Basically similar to a epidermal papillae, a dermal papillae here. Of course, this is mucosa, this is submucosa, and then we have muscularis, the bundles of interlacing muscle present on the far left.